I welcome everyone to my lecture. I'm Ms.T.R.Sambavi, Assistant Professor from Department of Industrial Biotechnology. So I'm here to take Enzyme Engineering and Technology subject. My today's topic is a general introduction to enzymes. So, introduction. Life depends on series of chemical reactions. We say this, every living organisms have to undergo certain series of reactions. So to maintain a balance in all the systems that is present in a body. So we highlight here about chemical reactions. This is because this topic is specifically about enzymes. So all the chemical reactions that is taking place within the living organisms, they take their own time frame for the reaction to take place. So all these time frame can be controlled by a thing called enzymes. So this topic is all about enzymes. So I highlighting all the chemical reactions that are taking place within the living organisms. So chemical reactions are far slow to maintain life. Living system has designed catalysts to fasten the specific reactions. So as we said, these chemical reactions, they are far slow to maintain life. So in order to fasten the reaction, or in order to reduce the time frame for that particular reaction to happen, nature have designed this catalyst which is used to fasten any specific reactions. Most essential reactions are catalyzed by proteins called enzymes. So there is a general talk that all the enzymes are a class of proteins. Okay, so say for example, we are talking about the chemical reactions. Say for example, we'll take this digestion process within our body. So every living organisms have food for their survival. So once they consume this food, it enters into the digestion system where this food is digested into other nutrients. So these food particles are broken down into several fragments, and these fragments are broken down into the required nutrients. So all these nutrients have to maintain the process that are taking place in our body, so that it helps the organisms to function its own functions and all. So it makes the organs to function. So these processes are also facilitated by enzymes. There are a lot of enzymes that are involved in this process. Then what is catalyst? So okay, catalyst speeds up or slows down a chemical reaction but remains chemically unchanged itself. So we say this catalyst, they are here to speed up or slow down a chemical reaction. Say for example, this A and B reactants, okay, they say let them take these reactants A and B. These reactants are giving a product C and D. So how this catalyst will influence the particular reaction? So they'll create a pathway to slow down the or to slow down the time frame. Okay, the particular time frame, it speeds up the reaction. It creates its own pathway and make the product. So from A and B reactants, we get C and D product. So it creates its own pathway. And we have to understand one thing here. This catalyst is not fully used up in the reaction. It is only going to facilitate. So that is why we are telling it remains unchanged. Okay, it remains chemically unchanged. It is not directly taking part in the reaction. It is only creating a pathway for the reactants to form the products. That is, it is just increasing the speed of the reaction. So say, for example, we'll take exothermic reaction. What are exothermic reactions? So in any reaction, so A and B, these reactants are forming a product called C and D. So heat is generated up in the reaction. So we call them as exothermic reaction. So if we increase the heat in this reaction, the yield will be low. That is, if we increase the temperature in the reaction in order to get the C and D product within a short time frame. So if you are going to increase the temperature in this reaction, the yield is going to be very low. So this is where we will use catalyst. So catalyst will create its own pathway for the reaction and will facilitate the reactants to form its products in a short period. So it increases the speed of the reaction. To be more precise, I have mentioned here about contact process, that is H2SO4 production, where it is used in detergent industry. The major use will be in detergent industry. Here, vanadium oxide or vanadium pentoxide, okay, it is called a catalyst. It acts as a catalyst in this reaction. So from sulfur oxide and oxygen, we are going to get this H2SO4. So this vanadium oxide or vanadium pentoxide is going to be the catalyst for contact process. So likewise, there are a lot of processes, chemical processes, which are catalyzed using a catalyst. So there are a wide range of catalysts are used for different chemical reactions to speed up the chemical reaction. So a catalyst works by lowering the activation energy for the reaction. Let us see how a catalyst works. All these time I was telling a catalyst is going to create a new pathway for the reactants to form a product. We'll see how exactly this catalyst is working on the reaction to speed up the reaction time. So if we see the difference, I have shown two different reactions here. One reaction is using catalyst and the other reaction is not using catalyst. So if you see the formation of the product all the way from the reactants to the forming a product, the reaction where we have used catalyzed will have very low activation energy when compared to the reaction that is where we are not using any catalyst, uncatalyzed, say, let us say uncatalyzed reaction. Fine. This is because whenever the activation energy is getting lowered, the free molecules, all the free molecules will immediately get converted into the product. So what is this activation energy? Why do we need to lower the activation energy? Okay, the energy needed to break the bonds in the reactants before any reaction can take place. So let us take an example where formation of water, H2O, from hydrogen and oxygen, when the activation energy, this activation energy is required for breaking the HH bond. Okay, when the activation energy lowers, this free hydrogen bonds will take the oxygen, and the formation of H2O will be faster when compared to an uncatalyzed formation of water reaction. So this catalyst basically lowers the activation energy for any reaction, and it speeds up the it increases the time of the reaction. Okay, so this is how a catalyst acts. So let us see the historical highlights of enzymes. So how they have discovered enzyme and who named the enzyme. So in 1700s, enzymes have the concept of enzyme emerged from this year. So how this concept have emerged? Digestion of meat by secretion of the stomach. So initially I gave digestion process as an example. Okay, so here exactly digestion of meat by the secretions of stomach. What are all those secretions? They found like some secretions from the stomach are digesting the food that we are taking in. Okay, someone have studied that how the food particles, where the food particles are going, it have been converted into the required nutrients by several secretions from the stomach. So in 1800s, conversion of starch to sugar by saliva and plant extracts from several plant extracts, and by saliva, conversion of starch into sugar have taken place. So all these discoveries were the base for a concept called enzyme to emerge. Louis Pasteur in 1850s, he have witnessed the fermentation of sugar into alcohol by yeast is catalyzed by ferments. That is like for getting alcohol, fermentation of sugar the end product is alcohol exactly. So Louis Pasteur have witnessed this particular reaction, and he called them as ferments. So they are catalyzed by a thing called ferments. Ferments are inseparable from living yeast cells. Fine. So he only discovered this fermentation process. So he was giving a base for conversion of sugar into alcohol, where this conversion have got into advanced stage. We are producing ethanol from certain sugars. A lot of bio wastes and a lot of bio waste streams are used for converting the sugars into alcohol. Say, for example, let us take this waste from all fruit and vegetable markets. Instead of dumping all these fruits and vegetable into and they just incinerate it to dispose that waste. So people have started utilizing that waste to produce ethanol. So the Louis Pasteur have initiated this fermentation of sugar into alcohol. Now we are having an advanced technology to get ethanol. So we call them bioethanol. So this is how the process have been advanced, and they have discovered the thing called enzyme. So Buchner in 1897, yeast extract could ferment sugars to alcohol. He also witnessed the same which was done by Louis Pasteur. So fermentation was promoted by molecules that continue to function when removed from cells. So he also witnessed the fermentation process. So he said, like certain molecules are responsible for this conversion. Okay, so Louis Pasteur just identified like ferments. He identified like something is catalyzing the fermentation process, and he called them as ferments. And now later, another scientist have witnessed the same, like a fermentation of sugar into alcohol, and now he have given an advanced term called molecules. Some molecules are responsible for this conversion process. And later, Frederick he called those molecules as enzymes. The molecules that have been discovered by Buchner, he said like certain molecules are responsible for the conversion of sugars to alcohol. Later, Frederick, he said that these molecules are called enzymes. So this is how enzyme got its name. Fine. So later, James, he in 1926, he did the isolation and crystallization of urease. So urease enzyme have been isolated and crystallized. So he postulated that all enzymes are proteins. So initially, if we look into the history of enzyme, someone have witnessed a conversion of sugar into alcohol, and the later the scientist who witnessed the same process, and he said some molecules are responsible for this conversion, and without naming this we can't go forward. So later the other scientist, he said the molecule that is responsible for converting sugar into alcohol, let us call them as enzymes. And later another scientist came and gave an advancement. So he isolated and crystallized the urease, and he said like all the enzymes are proteins. Fine. So John and Moses in 1930s, they crystallized pepsin, trypsin, and other digestive enzymes. So this is how like a discovery has got its advancement, and now we have advanced process and technologies for the same concepts. So all found to be protein. So whatever he crystallized, like they have crystallized pepsin, trypsin, and other digestive enzymes. So they said like all are found to be proteins. So they have they have just supported the postulates from James. And later one scientist, he wrote enzymes, weak bonding interactions between an enzyme and its substrate might be used to catalyze a reaction. So he exactly said like a weak bonding interaction between an enzyme and its substrate might be used to catalyze a reaction. He just found how this particular molecule called enzymes are facilitating the reaction to take place very soon. So a weak bonding interaction is there between the enzyme and the substrate. Fine. So then in later in 20th century, the purification and elucidation of the structure and mechanism of many enzymes have happened. So after the discovery, right now we are like producing an enzyme. We purify them partially, and we go till the application of that particular enzyme. So most enzymes are proteins. So a lot of scientists have postulated like all the enzymes are proteins. Okay, with the exception of a small group of catalytic RNA and DNA molecules, all enzymes are proteins. So with the exception, now we can tell more precisely, like except a small group of catalytic RNA and DNA molecule, as all the enzymes are proteins. The catalytic activity depends on the integrity of the native protein conformation. Okay, we are telling this catalytic activity, like how the enzyme facilitates the reaction to take place much faster. It depends upon their native protein conformation. So enzymes have molecular weights ranging from 12,000 to more than 1 million. Okay, so they more precisely, like a lot of postulates have been listed here about proteins. Okay, proteins and enzymes. Some enzymes require no chemical groups for activity. Other require an additional chemical component called a cofactor or a coenzyme, which is a complex organic or metalorganic molecule. So I will talk about this cofactor and coenzymes in the upcoming slides. So these are all the general points about enzymes and proteins, how these enzymes and proteins are related. Fine. Okay, so enzymes are biomolecules that increase the rate of the chemical reactions. So what are the enzymes? What are enzymes? Now, they are nothing but biomolecules. They are general biomolecules which can increase the time of the reaction. Like it will make the reaction to take place in the short frame. And also they increase the rate of the reactions. We can tell like they can increase the rate of the chemical reaction. And almost all the enzymes are proteins. In enzymatic reactions, the molecules at the beginning of the process are called substrate. What are substrate? So we are telling like enzymes are facilitating a chemical reaction. So before getting into the product, a particular molecule is going to be taking part in the reaction. So at initial stage, before getting into the process, those molecules are all called as substrates, and the enzyme converts them into different molecules. They are called as products. So from substrate, products are formed, and this reaction is facilitated by a enzyme. That is, the rate of the reaction is increased for the substrate to get converted into a product. So enzyme reactions. So any substrate is there. So we are targeting this enzyme. So enzyme plus substrate, it forms, we call it as enzyme substrate complex. Once this enzyme go and bind into the active sites, so it is called as the enzyme substrate complex. And this enzyme substrate complex will form product. So this enzyme will get detached from the this complex is getting dissociated into product, where this enzyme is getting detached totally from the product. So you will get a enzyme plus product at the end. Fine. So this is how we'll get the product from a substrate. Okay, this is to be more precise, it is an basic representation of a enzymatic reaction, how a product is formed from substrate with the help of the enzyme. So a reaction pathway. So we can see like the reaction, graph between reaction coordinate and the free energy. So the reactants, they'll undergo a transition state, that is a transformation of the reactants into products the activation, look at the activation energy. Chemical reactions need an initial input of energy called activation energy. Like I have given the formation of water as a best example for the process. So the bond between hydrogen and hydrogen need to be broken down. So for that we need activation energy. So this activation energy will break is the amount of energy that is required to break that hydrogen-hydrogen bond, so that this hydrogen molecule will go and bound to oxygen. So it will be easy for the hydrogen molecule to go and bind to the oxygen. This is how the reaction is represented graphically. Okay, during this part of the reaction, the molecules are said to be in a transition state. Okay, they during the when reaction is taking place, the molecules, that is the in the substrate, the molecules are said to be in transition state because they are going to be get converted into products. So this is the general representation of reaction pathway. All right. Now, an enzyme controlled pathway. If you look into the enzyme controlled pathway, the previous one was without any enzyme. The previous general enzymatic representation, we have graphically represented here. It is without a catalyst. So let us compare a reaction where we have used catalyst and we have not used any catalyst. So the activation energy required for a reaction using catalyst will be very low. So basically if we use enzyme, the activation energy is lowered and the products are formed. So enzyme controlled reaction proceed 108 to 1011 times faster than corresponding non-enzymatic reactions. So let us see the enzyme name rule, how we could name a particular enzyme. Let us see the naming rule. Enzymes are named by the substrate acted upon by the type of reaction catalyzed by adding ase. Example, cellulase, protease, amylase, lipase, glucose oxidase, DNA polymerase. So the basic naming rule for enzyme is based on the substrate that the enzyme is going to act or the type of the reaction that they are going to facilitate. The first name of the enzyme will be based on either the substrate that they are going to act or the type of the chemical reaction that they are going to catalyze. And then the second part of the name of the enzyme will be ase. It is a common name for all enzymes. So it is a common name. The common name for the second name of the enzyme is commonly ase. Say for example, cellulase. Why cellulase? Because cellulase are going to act on cellulose to convert them into glucose molecules. So the substrate is going to be your cellulose here, and the enzyme is cellulase. So where they are going to convert them into glucose. Likewise, protease, amylase, lipase, glucose oxidase, and DNA polymerase, etc. So let us see the enzyme classification now. The enzymes are classified using different categories. So first, let us take the location of action. So where the enzyme action is going to happen. First, based on the location of action, the enzyme is classified into endoenzyme and exoenzyme. So let us take this endoenzyme. They are intracellular, as the name is suggesting. In endo, the intracellular, most enzymes of the metabolic pathways. So the enzymes that are involved in the metabolic pathways, they are endoenzymes. Exoenzymes, extracellular, break down large food molecules or harmful chemical. Example, cellulase. So previously we have mentioned cellulase. Why they are calling it as exoenzyme is they are going to break the external bond. So say the cellulose is composed of more than 10,000 glucose units, isn't it? So they are going to break the external beta glucose bonds in the cellulose. So they come under this exoenzyme category. So based on the location of action, enzymes are converted into endoenzyme and exoenzyme. And the second category will be presence in cell. So constitutive enzymes synthesized by the cell in the absence of any specific stimulus always present in cell regardless of the amount of substrate. Enzymes that are always present and active. So you don't have to give any precursor, or you don't have to go induce it. So they come under this constitutive enzyme. So inducible enzymes, enzymes whose synthesis is stimulated in the presence of a chemical or physical stimulus. No, physical stimulus in the sense this heat, light, and all. So they'll act only with this physical stimulus, not constantly present in cell, and is produced only when the stimulus is present. Enzymes that are synthesized or activated when needed. So this constitutive enzymes are always present within the cell. But these inducible enzymes, they are present in the cells only when there is a necessity of them, only when the reaction takes place and this enzyme is required there, these enzymes are present at that time. That's where we tell enzymes that are synthesized or activated when needed. Prevent a cell from wasting energy by making enzymes that will not be used immediately. So only when the requirement of that particular enzyme is there, the enzyme will be present in the cell. And the third category is main category, it's a biochemical action. This biochemical action is what kind of reaction this enzyme is going to facilitate. Based on that, they are classified into 6 classes: oxidoreductase, transferase, hydrolase, lyase, isomerase, and ligase. So this 6 classes, depending upon the nature of the reaction catalyzed. So oxidoreductase, redox reaction, transfer of electron or hydrogen from one compound to another. So this oxidoreductase enzyme is basically we term it with redox reaction. Okay, so they transfer electron or hydrogen from one compound to another. Transferase, transfer functional groups from one substrate to another. So that is why they are called transferase. And hydrolase, cleaves bonds of molecules with addition of water. So to any reaction, it cleaves the bond by, and the addition of water takes place. So they are called hydrolase. And lyase, add or remove functional group from double bonded substrates. So the difference between this transferase and this lyase is transferase will transfer the functional groups from one substrate to another, and this lyase add or remove functional group from double bonded substrate. So we need to differentiate this transferase and lyase. Okay, where exactly this lyase is acting and transferase. So remember double bonded substrate. Okay, in case of lyase. Then isomerase, change substrate into its isomeric form. Okay, it moreover deals with the isomers. And ligase, formation of bonds so substrates can bind together, also known as synthetases, need input of ATP and removal of H2O. So it needs input of some energy, this ligase class of enzymes. So based on the biochemical action, the enzymes are classified into 6 classes. What kind of reaction they are going to catalyze. Fine. So the enzyme commission number. So for each and every enzyme, this enzyme commission, they have given a unique number for each and every enzyme. A numerical classification scheme for enzymes based on the chemical reactions they catalyze. So based on the chemical reaction and the nature of the enzyme, so this enzyme commission have given a unique number for each and every enzyme. Fine. So on what basis they have given this number? Let us see now. Enzyme commission number system. So this enzyme commission number, EC number for each and every enzymes, it is based on the what kind of class the enzyme is going to act on in other details. It is basically 4-digit number. First number denotes the class of the enzyme. So we have seen the class of the enzymes, isn't it? Based on the biochemical action, all 6 classes are there. And the second number is based on the subclass by the type of substrate or the bond that is cleaved. Okay, based on the bond that is been cleaved, it depends upon that. And Third number is subclass by the electron acceptor or the type of the group removed. So based on the electron acceptor or what kind of group is being removed, because either we'll add an um, bond to it, or we are going to remove some bond from the reaction. So that is how a reaction is taking place. So based on that subclass, this third number is given. And the fourth number, serial number of enzyme found. So classification based on the chemical reaction catalyzed, not on the source of the enzyme. It is based on the classification of the chemical reaction. It is not based on the source of the particular enzyme, uh, from where the enzyme is going to come. So enzyme activity, how to define an enzyme activity is, it is defined as the amount of enzyme that will convert a certain amount of substrate to product in a specified period of time under conditions of constant temperature and pressure. So how to define an enzyme activity is the amount of enzyme, let us say, for example, let us take I have 5 gram of cellulose. So I am going to convert the cellulose into glucose molecules. Fine. So let us add some 5. It is just a rough. I'm just giving a rough example. So for this 5 gram of cellulose, I am going to add 5 ml of cellulase enzyme to it. So okay. So I'm going to see how many the amount of product that is formed from the amount of substrate. Okay, so the how efficiently my enzyme is converting this substrate into a product under and I'm going to see the following parameters also, that is your time period taken for the enzyme to convert the substrate into product under conditions like at what temperature it is acting and what is the pH that is been used for the conversion of substrate into product. So this is a basic definition for enzyme activity. So how you are going to denote your enzyme activity? It is based on the international unit. What is this international unit? This international unit is defined as the amount of enzyme that can convert 1 micromole of substrate into product per minute at 25 degrees Celsius. So this enzyme activity and definition for an international unit is more or less similar, where we are going to more precisely tell like micromole, 1 micromole of the amount of enzyme that is converting 1 micromole of substrate into product at 25 degrees Celsius per minute. So we have kind of refining few parameters here in case of international unit. And Katal, it is defined as the number of moles of substrate transformed into product per second at 25 degrees Celsius. So these are all like different this international unit is said to be a standard way of representing your enzyme activity. Likewise, there are several representation for the enzyme activity, but majorly all research groups and the most acceptable form is your international unit. What is specific activity? Specific activity is defined as the number of enzyme units per milligram of protein, that is your micromole per minute per mg of protein. Specific activity. So the specific activity is nothing but the number of enzyme units that are acting for the conversion of substrate into product. Okay, this is valuable during enzyme purification. So before purification, the concentrate, okay, let us take like I'm going to partially purify my enzyme using let us say I'm going to purify using tangential filtration flow. So after purifying your enzyme, you are going to denote the activity of the enzyme, the specific activity. Okay, you're going to denote the activity of your enzyme as specific activity. As enzymes will become more pure and they'll be more specific in their action. So that is why we have going to denote this enzyme in specific, in terms of specific activity once they are purified. So thank you. That's all for today's topic. We'll talk about the production, purification, and other strategies for purification strategies of enzyme in the next class.